Hi, I'm Assam, the lecturer here at the Institute of Medical Education, and today we're going to take a look at solutions to Section 3 of ACER's GAMSAT Pink Booklet, Practice Test 3, specifically Unit 38, questions 106 to 108. And in this unit, we're going to take a look at optogenetics. Now, ACER's being a bit cruel. They haven't given you a diagram or figure of how optogenetics works, and I did that on purpose because if they did give you the figure and diagram, answering the questions would be a breeze. But I think it's important to understand the significance of optogenetics, and they kind of do touch on it in the stimulus. But the importance here is we can actually turn on and off processes in the brain using optogenetics. So that's the big. It's insane in terms of, like, for example, let's say Alzheimer's or Parkinson's or any types of neurological diseases. If we can play around with turning on and off certain regions in the brain, that'd be incredible. So how does it work? So they've given us a couple of examples of channel proteins such as CHR2. This was found in algae, and what we're told is if you shine blue light on this, let's say this protein or this channel, you can control sodium ions passing through the cell membrane. Now, they don't talk about it specifically in the unit stimulus, but it's important to note that this does relate big time with the brain, which is why I've kind of drawn it here, the diagram where we're injecting the virus with our gene and promoter into the mammal brain to activate or to inhibit, let's say, our neurons. So we're told that the gene for such light-sensitive proteins, they can be inserted into mammal cells. So first we have to splice the gene and the promoter together. We put them into a vector, such as a virus. Then we can inject them into, say, for example, here the mammal brain. Then we can shine blue light to either activate or inhibit the activation of the channel. So what I've drawn here, it's important to note, you have to make sure that if you're going to splice the gene from this other organism, you have to keep the promoter, because the promoter has to be specific for a particular mammalian cell type. It says that in paragraph 3 of the unit stimulus. If you don't have the promoter region, you're not. So the promoter region tells the cell to, say, upregulate this gene. If you don't have the promoter region, you're not going to increase transcription of this gene. So the promoter region is essential. And you've got to make sure you understand as well, promoter regions can be specific for certain cell types. If you have a promoter region that's not specific for the cell type you're putting it into, so if this promoter region wasn't specific for the mouse, the neurons in the mouse, it wouldn't work. So you wouldn't get this transcription of the gene. So the promoter and the gene have to be together, and the promoter has to be compatible for the mammalian cell type you're putting it inside. So that's important. So what we do is we put it into a virus, and then just a vector. So it's obviously in a virus. It's not like it's gonna do any harm. But you inject it, for example, here in the mammal brain. So we've got a mouse, and then when we put it into the mammal brain, they don't say this specifically, but you have to understand the gene. So the promoter then facilitates transcription of the gene, and then the gene can then get translated into the protein, which gets inserted here into the neuron, which can then get manipulated by blue light to facilitate sodium ions and, say, action potentials in this part of the region of the brain to facilitate activation of a certain part of the brain, or you can kind of inhibit this blue light to inhibit the depolarization or membrane potential. So that's the theory behind it. Now, let's take a look at question 106. It says, how does the presence of light-sensitive channel protein in the cell membrane activate a neuron? So this, you kind of do have to have some sort of prerequisite knowledge in physiology in terms of how we activate neurons. So remember, action potential, depolarization, hyperpolarization. So when we depolarize a membrane or depolarize, say, a neuron to cause an action potential, we facilitate sodium ions into the cell. It increases the potential of the cell, so it kind of excites the neuron. If we want to stop the action potential, we can open up our potassium channels to release potassium, or we can open up chlorine channels to take chlorine into the cell to hyperpolarize the cell. So all of this, I guess, prerequisite knowledge about activation of neurons If you knew that, fine, you would have answered this in a breeze. But if you don't, I do suggest you go back and look into it. But the answer for 106 is obviously going to be A, because it allows for depolarization. If we depolarize the neuron, it's going to activate the neuron and it's going to have its effects. Because depolarization, we're facilitating sodium into the cell. If it was hyperpolarization, we'd be losing the sodium. So understand these terms. B is incorrect because the vector to enter the cell, that's not why the neuron's activated. C says it increases the production of synapses. That's way off the truth. It increases the storage of neurotransmitters in vesicles. No. So remember, again, all of these, if you knew that action potentials were generated because of depolarizing of the cell via the sodium channel opening, you'd know straight away that's the answer. So now if we move over to 107, it asks, after gene insertion leading to the introduction of light-sensitive channel protein into a mammal, which one of the following limits the cell types activated in response to light? Now, we know the answer of this. It says it there in the third paragraph of the unit. It says it right there in plain English. It says, together with a promoter specific for a particular mammalian cell type. So the answer for 107 has to be B. So we know it's B. So because if the promoter isn't specific for the cell type, you're not going to get the transcription. So the gene is not going to get transcribed. So it has to be specific. So just think of it this way. The promoter is the machinery for the promoter to work is specific for a specific cell type. So that's the best way to remember it. So that's why B, the specific promoter, is correct. Vector doesn't matter. You can use any kind of virus vector. The vector just tries to get the genetic material into the cell. So vector doesn't matter. You can use any type of vector. Size of the channel protein doesn't matter because if the promoter doesn't work, the size of the protein isn't going to matter. And brightness of light isn't going to limit the response because as long as any type of light activates the channel, it's going to open. So brightness wouldn't matter that much. So that's why the answer is going to be B, the specific promoter. So now if we go on to the last question, it asks, how does gene insertion lead to cell activity related to light? Now, we just got to understand what is happening here. So what we're doing is we're injecting the genetic sequence with a promoter, so in a virus, into the mammal, and then that gets transcribed. So that gets transcribed, and then that gets translated into the protein, which inserts into the membrane. So that's why the answer for 108 has to be, so the cell produces and incorporates the channel proteins, D. So that's what's happening. So metabolism, if we take a look, cell metabolism is generally accelerated. This has nothing to do with metabolism. I never know where they ripped that from. It says the vector distributes the channel proteins. That's incorrect. We're injecting the gene, the genetic material encoding for the protein. So that's incorrect. So it says specific enzyme production by cells is increased. Don't know what that means. So that's wrong. So the answer is D. What we're doing is we're putting in the genetic material. It gets transcribed and translated into the protein, and it incorporates into the membrane. So the answer is D. Now, with this unit, it did require some knowledge in genetics and how we transcribe and translate. Well, the central dogma of science, right? You have DNA, which just gets transcribed into your RNA, which then gets translated to protein. It's the same process here. If you did have a background in research, you would have known how to answer these questions straight away. I have a background in research, and when I saw this, I was like, oh, I'm familiar with this because I know a few people who actually work in the field of optogenetics. So again, if you didn't have a background, now you do. But if you do want to get more information about optogenetics, you actually just go on Google, or you can join a lab. I always do recommend people join research labs because it can help you not just in GAMSAT, but for life, to learn about what's going on in the world. But look, if you're still having trouble with this unit, you can post your comments or queries in the comment section below, or you can contact us directly. We'd love to help. Thanks for your time. Bye now.